Hello students, in the previous video we learned about plant growth and its growth rate. In this video let us understand about development of plants. So let us first learn what is differentiation, dedifferentiation and redifferentiation. Differentiation is basically a process in which the meristem cells and the cambium differentiate and mature to perform specific functions. So you can see this meristem has developed and differentiated to root cell, sieve cell and leaf cell. Now it cannot go back. In this, cell walls and protoplasm undergo structural changes. The capacity of cell division is lost. Example, loss of protoplasm to form tertiary elements. They also develop very strong elastic lignocellulosic secondary cell wall to carry water to long distances even under extreme tension. So, under certain conditions, the living differentiated cell regain their capacity of cell division. So, when this happens, we call it as dedifferentiation, like formation of meristems from differentiated parenchyma cells. Now, let us understand what is dedifferentiation. So, dedifferentiated cells can produce cells that again lose the capacity to divide but mature to perform specific function. So, this is called redifferentiation. Plant growth is open, that is, it can be intermediate or determinate. Differentiation is also open because cells tissues arising out of the same meristem have different structures at maturity. The final structure at maturity of the cell tissue is also determined by the location of the cell. Example, cells positioned away from the root apical meristem differentiate as root cap cells, while those pushed to the periphery mature as the epidermis. Now let us look at plant development. So it is a process which includes all changes in the life cycle of an organism right from the seed germination to senescence, that is when the plant ages. It is the sum of growth and differentiation. So we can represent it like this. First we have the meristematic cell which undergoes cell division, then it undergoes differentiation, and then we have a mature cell which finally dies. So the plants follow different pathways in response to environment or phases of life to form different kind of structures. This ability is called plasticity. Like heterophyly due to phases of life, like in cotton, coriander and larkspur, the leaves of the juvenile plants and mature plants are different in shape, as we can see here. There can be heterophyly due to environment also, like difference in shape of leaves produced in air and water, like we see in buttercup. Now let us study about different factors which control the development of the plant. So we studied about intrinsic factors. These are intracellular or intercellular, and the extrinsic factors are light, temperature, water, oxygen and nutrition. So all these will determine the growth of the plant. Apart from this, there are plant growth regulators or hormones. So these are simple molecules that regulate the plant growth. These are of two types. Plant growth promoter. So they are for growth promoting activities like cell division, cell enlargement, tropic growth, like auxin, gibberellin, cytokinin are plant hormones. Then there are inhibitors. So like the auxin, gibberellin, cytokinin promote the growth, there are certain hormones which inhibit the growth, like abscisic acid and ethylene will inhibit the plant growth. They will cause activities like dormancy and abscission. So this is in response to wounds or stresses of the biotic and abiotic factors. Ethylene will fit in either of the groups, but we largely know it as a growth inhibitor. So let us study about all these hormones in detail. Charles Darwin and his son Francis Darwin observed that the cleopatiles of canary grass responded to unilateral illumination by growing towards light. Okay, so we call this phototropism, where a plant grows towards the light always. So then it was concluded that the tip of the cleopatyle caused the bending of the entire cleopatyle. Then F.W. Wendt isolated auxin from the tips of these cleopatiles of the oat seedling. So this is how they came to the conclusion that auxin are produced on the growing apices of stems and roots from where they migrate to the region of their action. Auxin actually comes from a Greek word which means to grow. So it causes the plant to grow towards the sunlight. Because what happens is the auxin goes towards the shade and it causes cell elongation there. There are different types of auxin. They can be natural or synthetic. Examples of natural auxin would be indole 3 acetic acid, and for synthetic there can be naphthalene acetic acid. Now, the function of auxins are to initiate rooting in stem cuttings for plant propagation, to promote flowering, to prevent fruit and leaf drop at early stages, to promote the abscission of older leaves and fruit, to induce parthenocopy in tomatoes. They are also used as herbicides. Like for example, 2,4-D is used to kill dicot weed. It does not affect the monocot plant, so it is a very important herbicide, right? It also controls xylem differentiation and helps in cell division. In higher plants, the growing apical bud inhibits the growth of lateral buds. So this is called apical dominance. We see apical dominance in this plant where the apical bud is developed more. Removal of shoot tip results in the growth of lateral buds. So it is applied in tea plantation and in hedge making. Then we have gibberellin. So these are acidic plant growth regulators. E. corsava treated the stolid filtrates of Gibberella, which is a fungus, to healthy rice seedling. As a result, it shows symptoms of Bacani disease. So later, the active substance were identified as gibberellic acid. You can see here the plant has Bacani infection, right? Here we see elongation and here we see stunting of the rice plant. There are more than 100 gibberellins in fungi and higher plants. Gibberellic acid is one of the first discovered and most intensively studied gibberellins till date. They cause increase in length of axis, so they are used to increase the length of grape stalk, to elongate and improve the shape of fruits like apples. They delay senescence, so the fruit can be left on the tree to extend the market period. They are also used to speed up the malting process in brewing industries. Sugarcane stores sugar in stems. So spraying the sugarcane crop with gibberellin increases the length of the stem. It increases the yield by almost 20 tons per acre. Spraying juvenile conifers with gibberellin hastens the maturity period. It leads to early seed production. So for bolting in wheat, cabbages and many plants with rosette habits, they are used. Now let us study about cytokines. F. Skoog and co-workers observed that from internodal segments of tobacco stems, the callus proliferated only if nutrient medium was supplemented with extracts of vascular tissue, yeast extract, coconut milk or DNA. Skoog and Miller later identified and crystallized the active substance and termed it as kinetin. So cytokinins were discovered as kinetins from the autoclaved herring sperm of the DNA. Kinetin does not occur naturally in the plants. Zeatin, which we get from coconut milk and corn kernels, is the natural substance with cytokinin-like activities. These are some synthetic compounds with cell division promoting the activity. Natural cytokinins are synthesized in region of rapid cell division like root apices, shoot buds, young fruits, etc. So what are their functions? Their functions are that they play a role in cytokinesis, they help produce new leaves, chloroplast in leaves, lateral shoot growth and adventitious shoot formation. They help overcome the apical dominance. They promote nutrient metabolism which helps in delay of senescence. Now let us study about ethylene. Auxins confirm the release of volatile substance from ripened oranges that hasten the ripening of stored bananas. Later, this substance was identified as ethylene. So ethylene is a simple gaseous plant growth regulator. It is synthesized in large amount by tissues undergoing senescence and ripening fruits. Let us look at its function. It influences the horizontal growth of seedlings, swelling of axis, and apical hook formation in dicot seedlings. It promotes senescence and abscission of plant organs, especially of leaves and flowers. It promotes fruit ripening. It enhances respiration rate during fruit ripening. This is called respiratory climatic. It breaks seeds and bud dormancy, initiates germination in peanut seeds, sprouting of potato tubers. It also promotes rapid internode elongation in deep water rice plants. It helps leaves and upper parts of the shoot to remain above water. It promotes root growth and root hair formation. This increases the absorption surface. It is used to initiate flowering and for synchronizing fruit set in pineapples. It also induces flowering in mango. It is widely used in agriculture. Now, the most widely used source of ethylene is Ethiphone. Ethiphone is an aqueous solution readily absorbed and transported within plant, and it releases ethylene very slowly. Ethiphone hastens the fruit ripening in tomatoes and apples and accelerates abscission in flowers and fruits. It promotes female flowers in cucumbers, thereby increasing their yield. During mid-1960s, it was reported that three kinds of inhibitors, inhibitor B, abscission 2, and dormin, were found in plants. They were chemically identical and now we call them as abscisic acid. So ABA or abscisic acid is derivative of carotenoids. It regulates abscission and dormancy. So let us study their functions. It acts as an inhibitor of plant growth and metabolism. It inhibits seed germination. It stimulates closure of stomata in the epidermis. It increases the tolerance of plants to various kinds of stresses. Therefore, it is also called as a stress hormone. It has an important role in seed development, maturation, and dormancy. Seed dormancy by abscisic acid helps to withstand desiccation and other factors and other unfavorable conditions for growth. Now, let us try and understand the interaction of all these plant hormones. So, these hormones play individualistic as well as a combined role in the growth. Such roles may be complementary or antagonistic. Antagonistic means they may cut each other's action out. The plant growth regulators interact to affect dormancy in seeds, abscission, flowering, senescence, vernalization, apical dominance, seed germination, plant movement, etc. In most cases, abscisic acid acts as an antagonistic substance to gibberellins. There are various factors which influence the action of these plant growth hormones. There can be intrinsic or extrinsic factors. The extrinsic factors include light and temperature. Now let us study the role of light and temperature on the flowering plant. So it is basically the response of plant to day or night. Some plants require light to induce flowering. Based on the light duration, the plants can be put into three groups. They are long day plants, short day plants, and day neutral plants. Long day plants require exposure of to light for a period exceeding a well-defined critical duration. Short day plants require exposure to light for a period less than the critical duration before the flowering is initiated. Day neutral plants have no correlation between exposure to light duration and the induction of flowering. So while shoot apices modify into flowering apices, they cannot perceive photoperiods. So the site of perception of light dark duration is the leaves. It has been hypothesized that there is a hormone for flowering. When plants get enough photoperiod, the hormone migrates from the leaf to the shoot apices, and this is what induces flowering. Now let us look at what is vernalization. So it is a phenomenon in which some plants depend quantitatively or qualitatively on exposure of low temperature for flowering. It gives several advantages. Like for example, it prevents precarious reproductive development late in growing season. It enables the plant to have sufficient time to reach maturity. Examples of vernalization can be seen in some food plants like wheat, barley, rye. They have two varieties, spring varieties and winter varieties. So spring varieties are normally planted in the spring and they come to flower and produce the grain before the end of the growing season. Whereas winter varieties, if they are planted in spring, they would normally fail to flower or to produce mature grain within a span of flowering season. Hence, they are planted in autumn, they germinate, and over winter they come out as small seedlings. They resume growth in the spring and are harvested usually around midsummer. Now let us look at some examples of this phenomenon. So this is generally seen in biennial plants. Biennials are monocarpic plants that normally flower and die in the second season. Like for example, sugar beet, cabbages, carrot, etc. Subjecting the growth of the biennial plant to cold treatment stimulates a subsequent photoperiod which generates a flowering response. I hope you all understood this chapter properly. Thank you.